for Amal Handal. I just want to tell you how we proceed normally on these things. We will give Amal about 15 to 20 minutes to summarize what she has found and her thesis. Of course, details are in the thesis itself, but she will go through this in 15, 20 minutes. If you have questions, please save them till the end of that and write them down or something. And you can, of course, we're going to give her a hard time, so we're going to ask her a lot of tough questions. But anyways, after that, we will, is there somebody else who can admit? Okay, all right. And then what we will do is we will take questions from an internal examiner and external examiner. Just so that you know, for the record, I supervise the thesis, and we have one internal examiner, Dr. Rami Arafa from the Polytechnic University, because he's internal, because it's a joint master program between the Polytechnic and Bethlehem University. The external examiner is Dr. Walid Al-Basra from Al-Najaf University, who's interested in orchids. And there is with us Dr. Alfred Vogler, who is, of course, our collaborator, under which this work was done, funded by the Royal Society. So we're grateful to the Royal Society. We're grateful to the Natural History Museum in London, which helped build capacities here in Palestine. So we will take questions then from internal and external examination, and me, I may ask a few questions also. And then we'll open it up for the audience that are here, and we have a good audience here from the museum and from outside the museum online, who will have a chance to ask a couple of questions also. Hopefully all the questions will be then at the end of the hour. We ask you basically to leave the room. You and the internal examiner and the external examiner will then meet among ourselves. Based on our evaluation, we tell Amal what needs to be done, if there's any changes, passes, whatever. We fill out the forms for the university, and that's how it goes basically for these processes. Once again, I just want to thank everybody who helped Amal, and she will, I'm sure, thank the people who helped her. But also, I want to say that I'm very pleased with the stage we have reached in this. We have two defense theses, one for Amal today and one on Friday for Tamara on beetles, phylogeny, and both have found what I consider important findings which will help us in Palestine. But more important, not just about this project, but for the future, so that we can develop our capacities here in molecular biology. And we will have a long-term collaboration with Dr. Alfred, with Rami, and with Walid Al-Basha and others about these things. So with that, I turn it over to you, Amal, and I'll start my timer.
Great, thank you, Dr. Mazen. Thank you again for everyone present here with us in person and online. And thank you again for everybody who has helped me throughout this entire process. This has been a long journey, so thank you all. Okay, so the title of my research is A Molecular Re-Examination of Taxonomy of Orchidacea in Palestine. Just a little background on Orchidacea. Orchidacea is the second largest family of angiosperms, or flowering plants. It has more than 28,000 species and more than 850 genera. They are extremely popular for their beauty and multi-purpose uses. They are used as ornamental flowers to decorate both homes and gardens, and they are used in perfumery and aromatherapy for example, perfumes, shower gels, candles, essential oils, and much more. They are also used as a food source. The famous vanilla bean comes from the orchid Vanilla planifolia, and the famous sahlab that we drink in the winter is made from the tuberous roots of orchids, usually the genus Orchis. They're also used for their medicinal properties. People use them to fight off bacteria, viruses, fungi, as well as to protect against liver and brain damage and cancer, sometimes even too. We can see, because of their multi-purpose uses, they are one of the world's most threatened taxonomic group. The major threats include overcollection, illegal trade, habitat modification, altered land use, invasive species, and here in Palestine, Israeli colonization. Just a quick background on Palestine. Palestine has a very unique geographical location. As we can see in the photo on the right, it lies at the crossroad of 3 continents, Africa, Europe, and Asia. This has caused it to have diverse climates and habitats and biogeographical zones. So as we see in the picture on the right, it has 4 different biogeographical zones. This has caused Palestine to have more biodiversity than some countries many times its size. So we have very rich faunal and floral biodiversity. For plants, we have more in historical Palestine, we have more than 2,800 plant species, with 155 of them endemic. And within the West Bank, we have approximately 1,612 species, with more than 600 of them rare. So orchids in Palestine. In historical Palestine, we have 45 species of orchids, and 23 of these species are recorded within the West Bank. Just in 2021 alone, 3 new to science novel species were described in Palestine. There has been many research work on orchids of Palestine. The first one is this book published in 2019 by Asaf Shifman, The Wild Orchids of Israel. That same year, Banan al-Sheikh published a book. It's not specifically on orchids, but it's about flora of the West Bank, and it contains much data about orchids of the West Bank. And then in 2021, Carl Kreutz published this book, Orchids of Israel. And this is the book that the 3 new to science species were described in. And then there's this scientific article published by Professor Mazen and Julius Pal. This reviews the distribution of orchids within the West Bank over the years. So just a little about barcoding of plants, DNA barcoding of plants. So DNA barcoding, as the name entails, basically gives a barcode or an identification for plants using molecular techniques such as short sections of DNA. Achievements in this field for both fauna and flora, but specifically for plants, in the past 20 years have far exceeded the sum of the previous 200 years. So we now know a lot more about plants thanks to DNA barcoding. But in plants, it is far more complicated than it is in animals. So in animals, we have a universal barcode, a universal gene, CO1, cytochrome oxidase 1, that we can use it and we can have barcodes for, beetles, whatever we want. But for plants, unfortunately, this is not the case. We have to use multiple different ones. Most of the DNA plant barcodes are using chloroplast genome sequences, as we see here on the right, and nuclear barcodes. But usually combinations work best. We can use combinations of internal transcribed regions, chloroplast genes, mitochondrial genes, or intergenic spacers. The most preferable combination is having a coding, so a gene with a non-coding region, so like the ITS or the IGS. If we were to talk about barcodes available for orchids, there's a lot of data missing on that. As we said in the first slide, there are 28,000 orchid species accepted, but the Barcode of Life database only has 4,597 barcodes available for orchids. And these barcodes are from 36 different countries. And as we can see on the map on the right, none of which are from West Asia or North Africa. We can see no dots whatsoever in our area. So there's a big gap. So what is the research objective? Our research objective is to fill the gap of lack of molecular identification of orchids in Palestine. And that's not just true for Palestine, but it's also true for the entire Mediterranean region, as we saw. We would like to contribute to the Global GenBank database by providing sequences of endemic species, as well as Mediterranean species. And with the knowledge that we get from this research, we would like to support conservation of threatened and endangered orchid species. So what did we do? We, over the course of spring 2021 and 2022, we went on 19 different field trips to 19 different localities across the West Bank, as we see in the map. We have gathered 144 orchid specimens belonging to 19 species. Of these 144, we have chosen 53 specimens to be sequenced. These 53 contain one sample per species, as well as any other specimen that had a different morphology. For example, it was light in color, or it had a different morph, it looked different than what we are used to. As for the DNA extraction and the PCR, these were done at the PMNH laboratories here. For the homogenization of the samples, we used liquid nitrogen with a mortar and a pestle to homogenize the samples. And for the extraction, we used nucleus pen kits by Meshri Nagel for extraction. As for the PCR, as I said in an earlier slide, primers of barcodes are preferred to be a combination of coding and non-coding regions. So that's what we did. We chose 2 genes, as you see in red, MATK and RBCL, and 3 intergenic spacers. MATK is a gene that codes for matrix K protein, and RBCL is a gene that codes for a large subunit of ribulose biphosphate carboxylase. And the IGS regions are non-coding regions, so they are not conserved, and that means they have lots of variability within the taxa. So as for the Sanger sequencing was done at the Natural History Museum in London. Sequences were aligned using Clustal W, along with our 51 specimen. We added 47 reference orchid whole chloroplast sequences, as well as 2 outgroups, Iris and Lycoris, which both belong to the same order as Orchis, Aspergillus. We did alignment for each primer set individually, and then we combined all these 5 primer sets together, and we concatenated them into a supermatrix to create a tree containing all products of the 5 primer sets. This is what a section of the alignment of the concatenated alignment looks like. All 5 primers together were about 3,500 base pairs long. For the concatenated tree, we did a maximum likelihood tree with 1,000 bootstrap replicates using IQ tree. For our results, PCR amplification was successful for all 144 samples. Sequencing, on the other hand, was only successful for 51 samples of the 53 samples chosen for sequencing. There was a tube mix-up for specimen 32, and we had a sequencing failure for specimen 47. So if we look at this chromatogram at the bottom, we have 2 samples here. One I chose by random, P102. They both belong to the primer RBCL, gene RBCL. The bottom one, P102, is very clean, very nice. We can see each peak on its own. We can see clearly which one is an A, which one's a G, etc. Whereas if we look at P047, the specimen that failed, we didn't see that. It's multi-peaked, it's very noisy, it's very messy, so we decided to omit this sample from alignment. This is what the concatenated tree looks like. That contains all 5 primer regions, as well as our 51 specimen and the reference specimens. Our tree has 5 subfamilies, as we see on the left here. They are each color-coded. Our Palestinian specimen belongs to 2 subfamilies of these 5. One species only belongs to the subfamily Epindroidea, and that's Limnodorum abortivum. And the remaining 18 specimens belong to the subfamily Orchidoidea. So if we were just to analyze this tree very, very quickly, this tree behaved just like as we expected it to. Limidorum abortivum was clustered with the reference Limidorum abortivum that we downloaded from GeneBank, and it was clustered in the correct subfamily, so everything was perfect with that one. If we look at the blue down there, we have Orchis as a, you can't see my mouse, but we have in the top here, we have Orchis as a clade. It's a monophyletic clade, Orchis, and it has two different species. Anacamptis turned out to be paraphyletic in our tree. And then we have Ophrys here, which is monophyletic. And then we have the Neotina tridentata in its own clade, and then we have the Serapius leventina. We will discuss each one of these in details in the next few slides. So if we were to discuss Orchis, Serapius, and Anacamptis, there has been much debate about the Serapius and the Anacamptis genera in the past. They both belong to the Orchis genus. For many, many years. And recent studies have shown that actually Serapius and Anacamptis are both distinct genera from the Orchis genus. An interesting paper was a cytogenetics paper done on that. And it showed that Orchis had 42 chromosomes, whereas Serapius and Anacamptis both had 36 chromosomes. So this was a nice paper to kind of say that there should be a distinction between the Serapius, Anacamptis, and Orchis. And this was also shown in our study. We can see that Orchis clearly is its own monophyletic group, and Anacamptis clustered not with the Orchis, but on its own, and then the same goes for Serapius. So our data supports that finding. Another dilemma over the past few years is the Anacamptis papilionaceae. This species moved from genera to genera, genus to genus very often. Sometimes it's Anacamptis, sometimes it's Vermulina, sometimes it's Orchis. But our tree shows that this specimen, this species, clustered with two other Anacamptis species. So this supports the fact that Anacamptis papilionaceae belongs to the genus Anacamptis. Lastly, Anacamptis culina, to our surprise, did not group with the remaining three Anacamptis species. It grouped on its own, and there is also debate on that. Some other people got the same result that we did. Some people got Anacamptis to cluster with Anacamptis using ITS regions. Other studies got it to cluster on its own. So this genus, this species is still needs more studies to be done on it to kind of figure out which genus Anacamptis culina truly falls within. Another thing we concluded from our study is that morphological differences does not necessarily infer genetical differences. So if we look at this top picture over here, this is PO43. This is what a typical morphology of Orchis galilea looks like. If we can see that Orchis galilea looks like a stickman and has a clown shape. The labellum is divided into three lobes, and then in the middle there's like this small tooth. In the spring of 2021, we found this specimen on the bottom. It looks different than the specimen on the top. The lobes, the three lobes, look kind of webbed, and they are not forked all the way through. We do not see the legs of the stickman like we do in the top picture. So we gathered a specimen of that, and we were interested to see the results of that. And just to make note, in spring of 2022, so a whole year later, we went back to the same exact location, and we went to the same exact plant. I remember this plant very, very clearly. The same exact plant was growing normally, just like a typical morphology of Orchis galilea. So in our tree, this is what the PO79 looks like. It's grouped with the Orchis galilea just normally in the same clade. So it's not a it's not a different species, or it's not a subspecies or anything like that. It just had a different morphology. And it's important to note that ecological situations do have an effect on morphology. I mean, the light, the temperature, everything can affect morphology, but that does not necessarily mean it will affect genetical outcomes. If we are to look at this Ophrys, seem to be monophyletic in our tree, though there are some questions to be asked. Ophrys is one of the hardest species, genus to work with. All the papers agree to that. Many papers support the following two hypotheses about why the Ophrys monophyletic group has questions. The first hypothesis that scientists have agreed on is that this fuzziness can kind of be evidence of introgression or hybridization between the taxa. Ophrys is one of the most genera that has hybridization within itself. The other one could be due to morphological misidentification, because the Ophrys is the hardest genus to identify and distinguish morphologically. All the different species almost look identical. I mean, there are differences, but some species are almost identical that even orchid experts have hard times identifying them. And in Asaf Schiffman's book, there were two misidentified Ophrys species. So morphological misidentification of Ophrys could lead to this fuzziness in the phylogenetic trees of Ophrys. Another thing we noticed in this Ophrys monophyletic group is we have this very long branch in the middle. This branch actually belongs to the species that belong to the section Pseudo-Ophrys. Out of our entire Ophrys species, only two species belong to this section, Ophrys lutea and Ophrys omigifera. And as we can see, Ophrys lutea grouped together and Ophrys omigifera grouped in the same monophyletic group with them. We thought there could be an issue with our sequences or with our alignment. We went through the sequences, we went through the alignment. There is nothing unusual at all with this, with these specimens. But it's important to note that we did use a maximum likelihood tree, and with maximum likelihood trees, branch lengths represent the numbers of substitutions per site. And as the name infers, also entails, they're called pseudo-Ophrys. So they are like Ophrys, but they're not actually Ophrys. They're Ophrys. So they are different morphologically. It's very clear to see that these pseudo-Ophrys are different morphologically from the other Ophrys species. And then it's clear for us to see that genetically these pseudo-Ophrys species are different genetically from the remaining Ophrys species. Okay, and then lastly, in our discussion, the Ophrys palestina was described as a new species. Ophrys palestina looks exactly just like Ophrys adenitis. Asaf Schiffman, in his book, did not notice the difference between these two species. They look exactly the same. They only have ecological differences. So Ophrys adenitis it grows up to 40 centimeters tall. Its labellum, which is this, you can't see my mouse, but it's this middle part of the flower, is 9 to 12 millimeters wide when spread, and it flowers from late January to mid-April. Ophrys palestina, on the other hand, that Carl Kreutz described in 2021, grows up to 50 centimeters tall. Its labellum is from 16 to 22 millimeters wide, and it flowers 3 weeks after Ophrys adenitis. And so we were certain that we had this species because we found all these morphological differences. We found two species that had the same description as Ophrys palestina. If we were to take P113 as an example, it grew up to 60 centimeters tall, and the labellum was 12 millimeters wide, and that's without it being spread. We anticipate it to be much wider when spread. We gathered it from April 8th. If we look at this phylogenetic tree, it is clear that there are two completely different species on the tree. Ophrys palestina P113 and P114 grouped together, whereas Ophrys adenitis all grouped together in their own clade. And if we notice here at the in the back, again, you can't see my mouse, but they both have different ancestors that they belong to within that group. Oh, that's good. Okay, so for our conclusion, this study provided significant new insights into the complicated taxonomic situation of orchids in Palestine. Our tree generated is in line with published work and adds new data. I'm not going to repeat these points that we just discussed in the discussion due to time limits, but I will talk about this slide, our last slide. We came up with many future actions and research that needs to be done based on this research that was done. First and foremost, these species that we sequenced need to be added to the gene bank. As we saw, there isn't any sequences from our region and from the entire Mediterranean orchids. It was very hard to find reference genes for the Mediterranean orchids. We also need an in-depth morphological and molecular study to be done on the Ophrys genera, and it's important to note morphological and molecular. Ophrys is a hard genus to work with. We need more detailed studies to be done relating to the resolution of the pseudo-Ophrys branch, as well as the Anacamptis choline branch. There are still questions to be answered about that. And then our study only used IGS and 2 genes. More studies need to be done that incorporate ITS regions and complete chloroplasts of orchids of the region to have an even richer molecular data of the orchids. And thank you. Any questions?
Thank you very much, Amal. It's very interesting.
Thank you, everybody. Now we open the for. Let's start with Dr. Walid and Dr. Rami. I don't know which of you wants to start. Let's start with Rami.
No, Rami, you are the one for molecular. For molecular, go ahead.
دكتور وليد خليني شوي ابدأ معها أنت يا دكتور.
Okay, Walid, you can start. We can always come back. We can have more than one round for each viewer.
In general, when I read the thesis, it's really prepared well, and for molecular diagnosis and for typing, it was really a great job and great work, which I cannot do any mention of any point regard what you did. But I have some general question about the work. For Amal, where was your part in this research? Because while you are talking about your research, you said we, our, until in your thesis, because it is a thesis, you should write yourself because it's your research. So where you have been within this research?
Yes.
As a paper, as a paper, you can write we, but for your thesis, it is your thesis. Where you been within this thesis?
Okay, thank you for this question. As Dr. Mazen said, this was a collaboration between the Palestine Museum of Natural History and the Natural History Museum of London. I began with this project ever since the first beginning of it. In the spring of 2021, I went on the first field trip to gather orchids from Palestine here. And I was on the field trips to gather the orchid specimen. I did the homogenization, DNA extraction, and PCR here at the Palestine Museum of Natural History. In the spring of 2022, I went over to London and worked with Dr. Alfred Vogler and Dr. Hoshi from the Natural History Museum in London, where the sequencing took place. And then when I came back, we continued working over online, Dr. Hoshi and I, Dr. Alfred and I, on the analysis on the generation of this concatenated tree. So
it's really important for me to hear that you participate in each part, because it's really important for your master thesis, because our audience should know that it's not you are part of each part of your research. So it's really important for us. The other point is the title. The title is called Palestine. are you sure it's Palestine, old Palestine, or a part? Because you miss Tubas, you miss another part, you miss the north part of Palestine as historical Palestine. Mainly, I was really disappointed with the map you put it in your presentation, because it's Israel, not Palestine, because it's including the Golan Heights, and you should not be including Golan Heights as Palestine. So I see Dr. Mazen smiling, because it is not Palestine, because we are representing for Palestine, and mainly you were on the West Bank.
Yes, that's a good point. I missed that. I apologize about that. Actually, when I was looking for the phytogeographical regions, I used to only find the West Bank, and I didn't just want the West Bank. At first, I just had a West Bank map with the phytogeographical regions, and I kept looking and looking for the for one that had the entire Palestine in it, and I found this one, and it missed me. I apologize about that.
It's not in the thesis. This is only in the poem.
Yeah. But just because the presentation, no problem. But for is it Palestine or West Bank? Because your work was in a part of the West Bank. It's not until the West Bank as a whole. Because Tubas area, I think maybe you if you worked within Tubas, and mainly for the the part of the Jordan Valley, maybe you will find more species, but you did not have any species from there.
We took a few field trips to the Jordan Valley area. There is one species there. We were not able to get it. We couldn't find it. Tubas معك حق يعني yeah, we will include it.
This is for future. This is for future, because really it's for me important. Another point, I look for the towns and the areas, like you talk about Haddad village area. Is it Haddad village area or Sab'in area, or it's Immetut Al-Marj? Which part?
It's actually the road right by the Haddad village. So we were in the car. We were looking for an Acamptis sancta, and it's very hard to find it. We couldn't find it much. And we found it on the road, like right next to Haddad village.
So it's Sab'in area. It's a Sab'in area. It's not Haddad. And for the Al-Jdaidi, is it in Nablus?
لا هذه نابلس الجديدة.
So because you write Al-Jdaidi in Sanur Valley, it's in Jenin.
Sorry, هاي يمكن typo, بس نابلس الجديدة.
No. I know all of us can make this, problem. And for Kana Valley, it's not Nablus. It's in Selfit. It's not in Kana. So you should. Kana Valley is in Selfit Governorate. It is not in Nablus.
Yes. شكرا على الملاحظة.
No, and in Arabic translation, everything you wrote Sahlabiyat, and once you say orchid. So take care of, because really, some points is really important but your work were really excellent. I learned a lot. Thank you for Dr. Mazen, because really it was really excellent work. And because I have many species, and Dr. Mazen told me to have, I already in the refrigerator, I have many species for him. Later on, I will send it to them. But you did a great job. Thank you so much for your work.
Thank you so much, Dr.
Thank you.
Okay.
Can you hear me?
Yes, we can hear you. Yes.
Okay, I would like first to thank you very much for this effort, Amal. It was really a challenging work, and I thought it would be it might be very problematic and difficult to proceed with such a work. You are working with a huge family, very diverse and very and distributed in so many places. So what a challenge! But you did it. You did very great job.
Thank you.
But that doesn't mean we can have some comments to improve the work and to make it looks better and somehow for the future to have a real nice output of it. Yeah. I would comment a few comments on the methodology and molecular work. And I have also other comments on the speciation story. I would start with the abstract, Amal. In the abstract, you mentioned that you collected 144 samples. And these 144 belongs to 19 species.
Yes.
So that means you represented some species with more than one sample, right?
Yes, okay, yes. So I did a minimum of one specimen per species, and then any confusing specimen that had different morph, like PO79 that I showed.
Do you have a population sample? I mean, the number of specimens or the number of samples in one locality, does it reach a good amount?
I added a new table in the most recent one that I sent. It's table number, I can look it up, but yeah, I added a new table for that. It's table 6.
So that would be very useful and helpful, Amal, to do a small population study examining the diversity inside the species. Inside the species. So this will help also to.
I would agree, Rami. This would be something that can be added to the recommendations for future population creation. Especially the Galilea situation would be an interesting situation.
Exactly. And this will also help very much in formulating a proper conservation strategy, especially for these species with a low population number. Did you collected the whole plant? You pull it whole or?
لا أكيد إحنا biodiversity center. لا we just cut the tip as we saw in the photos. Yes.
Okay, this is one comment. Another thing. Why? I hope you will answer this. Why you concentrated or chose all cpDNA markers? You have problem with ITS, for example, in a preliminary work. So you didn't include the nuclear genes. You only included a chloroplast genes.
Yes, I did much research about the different which genes to use and which genes not to use. The thing with the ITS regions, I mean, I read many papers. Most people for orchids use the IGS rather than the ITS. Yes. Right. And ITS in orchids have many pseudogenes, and it's therefore difficult. They didn't advise to use ITS much in orchids because it can get confusing because they have many pseudogenes within them. And the three most popular genes were the three that I chose. And there was a study done in Korea that tested a bunch of different genes for orchids, and these 5 were the best 5 that worked for them.
Have you came across any study with a coding region in the ribosomal DNA? Not the ITS, for example, 18 or 26S or 5.8S even. I mean, having a look at the paternal genome may also help a lot in understanding the speciation, the hybridization events. Maybe you could get some, let's say, conflict between the chloroplast and the nuclear DNA. But still, this is important information. Also, yes. This is, I think, need to be written in the recommendations, Amal.
It is written in the recommendation. I do think that is an important point. And I did include the last point of the recommendations is to include ITS as well as to include complete chloroplasts, because I saw that IGS and the genes are not sufficient for Palestine or for the Mediterranean region. It could have been sufficient for this study, but for this region, we need to do more ITS and more complete chloroplast and other genes also. Yes, thank you.
So, okay, thank you. I have a question. Why did you choose the irises as an outer group? Did you choose iris as outer group?
Yes, yes, and as I said,
an orchid from orchids around us. I mean, Chinese, European, African, whatever. Something
because they're still quite similar. I mean,
because choosing a very far taxa as an outer group may
yes. So the reference genes do contain orchids from China and from Korea and from different places. The reference genes, we added 47 reference orchid genes, and these are orchids from all over the world. They're not Mediterranean because we couldn't find Mediterranean complete chloroplasts. So these are in the reference genes. As I mentioned, the iris and the lycoris belong to the same order as orchids. So they are similar to orchids still. They're not too distinct. I mean, irises are pretty similar to orchids. So we wanted to choose something that was similar, but not too similar. And this was supported in other papers too, where they have used the iris and the lycoris as an outgroup as well.
Maybe I would disagree with this information that iris is close to orchids. Well, no, I don't think so. But anyway,
on an order level, we can't argue it.
Yeah, but you are working on the genera. I mean, you are comparing genera, so you don't choose an outgroup from another order. I mean, comparing or making phylogeny between genera, you choose a neighboring or a sister genera, maybe family. If you, I wouldn't go to the order. This is very, very far. Maybe you can try to
a little bit more on this issue of outgroups. Normally, when we select an outgroup for any group of species, we try to find things that are not too distant, because if you choose things that are too distant, the likelihood of terminal evolution and other problems. Sorry, can you hear me now?
Now, okay, I hear you. But I prefer that Amal will
when you choose an outgroup, it is best not to take a group that's too far out. But it's also not good to have a group that could potentially be an in-group, even though taxonomically, morphologically, they look distinct. So in this case, the iris is clearly separate, and the orchids cannot be confused for an orchid.
Okay,
but genetically, there have been other studies which show the relationship of these orders, and they show the orders of the iris and what's the other one? Yeah, that they are related. There is sequencing studies that show which used, when you look at orders like this, you use an even more distant outgroup. Of course, you're looking for order relationships.
This one initiated something else in my mind now. Are the subfamilies all represented in your tree, Amal?
Subfamilies. I only have 5 subfamilies. There's 6 subfamilies. I only have 5
subfamilies. Orchid subfamilies. Are they all included in your tree?
Yeah, there's 6 subfamilies. I only have 5 subfamilies in my tree.
Can you close the microphone? I can't hear you properly, Amal.
I said, no, they're not. There are 6 total orchids.
Okay, so choose one from the neighboring subfamily as an outgroup.
خلاص تمام هاي بنحط recommendation.
Okay. Well, let's go to the. You showed a very nice alignment, Amal. Can you go back to this alignment, please?
Yes.
The block, the big block. Because I noticed there is a gap, or there is.
Yes, this gap belongs to specimen.
Is it responsible for the long branch
La,
in
la,
the tree?
this, doctor, this.
Can you zoom more?
Type.
Yeah.
This
Okay.
gap belongs to P55,
P50, one species. Oh.
It belongs to PO55. PO55 is Serapis leventina, and as we saw from the tree, Serapis leventina is the most distinct one from all of the orchid specimen. So I noticed that also, and I looked into it, and it made sense that this gap, this deletion belongs to the Serapis leventina. We only have one sample of Serapis leventina.
Okay. And it has nothing to do with the long branch on the tree.
لا the long branch هم 72 specimen 72 هيا. 72.
Do they have something weird in the alignment? I mean, do they look like a separate block?
No, I went through the alignment, I went through the sequences, everything for these look normal.
Be careful. I'm skeptical a little bit, but...
Dr. Walid, do you have any more questions? Dr. Walid?
حط ميوت دكتور.
أنت حط mute. No, you're still mute. Unmute yourself. Yeah.
No, for Dr. Rami covering everything about molecular level because it's his specialty, and I'm interested in hearing all of that. And I don't have more question for Amal. You did a great job, really. It was really, until I told you, we got a good knowledge for, but for the reference for the neighboring country, it was, I was thinking of that while I'm reading, because we should need a control for what we are talking, because which is the original or which is the new species, I don't know, to have the reference for
The last point I want to discuss the geography.
because
you have to widen your lens according to the geography. You have to look at wider geographical scale, Amal, because we are a very tiny dot on the map.
Sorry,
so,
what I did.
As I
for the Orchis galili, the population which talk about Dr. Rami, about the population in one field, really in Nablus area, I am, this is for Dr. Mazen, although for the next Asira Shamaliya, there is a huge variety in colors in one place, and I think this is need a lot to do over, because I found one place like with 5 different colors in a small area, like not more than 1 meter square.
Could be
With
Dr. Walid, maybe. Could be a speciation event is running at the moment.
Dr. Walid,
very
we included
important.
many specimen that had different colors. For example, you know Orchis anatolica. You know how there's the white, completely white Orchis anatolica, and there's the purple Orchis anatolica. We actually sampled both of these species, exactly the same. Anacamptis papilionicea, some of them are very, very, very light pink. Some of them are very, very dark purple. We took both morphologies, and they all, and that's why, yes, that's why one of the titles was morphological.
Molecular level will judge this is something
Maybe,
intra-population variation, or is it a speciation event?
Could be. Can I give a chance to Dr. Elfried? Maybe you can give your intervention if you have any questions, Dr. Elfried, and then we'll go to the rest of the audience.
Well, I don't have an intervention really, but I enjoyed this. Thanks a lot, and I think it's came a long way from where we were even just a few weeks ago, so that's great to see. I mean, in terms of like what I can just think of is like so publishing these data. I mean, that should be probably high on your mind. Also, we need to do that for the output of our grant, so that's of some concern to me. but the one that kind of stands in the way is this long branch there, right? And so, I mean, although it was already discussed, I mean, I want to just go after this one more time. So why do you think that is, and what do you want to do about it?
Yeah, that was a very confusing branch for us, and we went through the morphology of these specimen again, and we compared them with the other ophis, and they clearly look different. I mean, sometimes you can distinguish between the ophis gene species, but these two, the Ophis lutea and the Ophis omegifera subspecies Israelitica, are different than the other ones, and they belong to this section pseudo-Ophis, and pseudo is yani fake, yani different. They are different. So
I mean, I have no doubts that these are distinct and somewhat unrelated to the other Ophis. So that's not a question. But
so we need to do more.
How do you get that? I mean, what can cause this kind of stuff? So let's say the alignment, as you established, that's not the issue. So what else is it? And what will you do?
Yeah, I think this is where more molecular work would need to come in, either doing the ITS or doing complete chloroplast if we are able to. And I know we already started working on that. Hushi already ran a few for the ITS and for the complete chloroplast. I think we would need more molecular data here to kind of distinguish what's going on there.
Well, that's always the easy way out, and that we don't have time and so on. We want to publish this now, let's say. So what about, I mean, what kind of models are you using, or what kind of tests do you have up your sleeve to establish, well, should that go there or not? Have you thought about this? Or is there some room? I mean, I don't want to put you on the spot here, but so, I mean, is there room to just play with, say, the models of molecular evolution that we would be able to apply here? Or is it, I guess, partly it would be somehow protein coding regions, so maybe that could be coded in a different way, right? And maybe another thought would be that we actually constrain it. I mean, do you know where it's supposed to go? So if you constrain Ophis as monophyletic, then that would leave that other group, the offending group, out, and would then find a place elsewhere and see if that is more meaningful. So maybe you can play with the conditions there a little bit.
If I may just add to this, I think we have enough data to publish a paper on this orchid data as they stand. Of course, it opened the door for a lot of other questions which are listed in the recommendations that Amal put for further work. And this will give us, and when Walid asked about why in many parts of the text we use the word we, because of course this will be published, and it will be a multi-author paper, including you, Alfred, and Hushi, and I on it, as this will be our first actual data here for molecular phylogeny of plants. It opens the door, as you pointed out, for potential further work. We have a call for proposals here from GEF, and I'm thinking of applying for some of this to continue the work, like what also Rami and Walid pointed out, some areas of population genetics, conservation aspects, use of orchids as conservation, covering areas like tubas and others mentioned by Walid. Collecting from the Galilee, there are some species there in the Galilee that we haven't had, and the species from the Sahara-Arabian and Sudanese elements that we don't have potentially. This opens up a whole avenue of research for us to pursue, and I'm really excited about these potentialities.
Yeah, I mean, I should say it's always great once you have like a framework there, then it's kind of easy to build on top of this, right? And so this is really like the big achievement now, Amal. I think that's great. And but you just need to tweak that. You need to get that branch down. I mean, that's just otherwise it's not going to go in any journal. So just think about
I'm
that.
always, when I take students like Amal or Tamara has tried the defense, or many of the students here that have master degrees, they know my condition for master acceptance is that they publish the paper. Because if it ain't published, it ain't worth a nickel, as they say.
It wouldn't work at Imperial, but
Yes.
yeah, I'm glad it works for you.
So, and this is being recorded, so Amal needs to commit here that he will publish this.
Okay.
Okay.
Guys, if you don't mind, I have to go to another meeting now instantaneously,
Thank you
so
very
I
much,
have to hang up. But really great seeing you and great to see the story unfolding.
Thank
Well
you so
done.
much.
Yeah.
Dr.
All right.
Thanks.
Bye-bye. Bye.
Bye-bye.
Bye.
Okay, other people either online or in person here want to ask questions, because I then have two questions to ask Amal, put her on the spot with them. So I'll reserve them till the end and see if they got asked or not. If not, I will. Anybody here in the audience in the room here? No? Come on, play the devil's advocate. Okay, on the audience online, anybody? Tamara, Dr. Isa. Okay, go ahead. Yeah. Why did you choose orchids? Good question.
Orchids for many reasons. The first reason is there was already 2 ongoing projects here at the Institute with orchids. There was the paper that was published in 2021 that I showed with Julius Pal. This was a bachelor thesis, and we were already working on orchids here in the Institute, and we already have an orchid ex situ conservation here at the Institute. But the second reason is because orchids are a fascinating family, and they are very beautiful to look at, very beautiful to work with, and we really enjoy them and we want to conserve them.
There's a third reason, if I may add. They are potentially a very good indicator for ecological decline of the environment, because they have specific pollinators. And if those specific pollinators are eliminated, like with pesticides, insecticides, then you can see the orchids disappearing. So the presence of orchids itself, and we've already seen them disappear from many parts of the West Bank. So they're like the canary in the mine, so to speak. They're a good indicator of the destruction of the environment. Okay, Dr. Waleed and Dr. Rami, do you have any other issues or questions to ask before I ask my questions?
I would have small recommendation, Amal, just to go sometimes for microscopic differences. You mentioned in one slide two totally different morphotypes, and you have in other case very similar but molecularly different, yeah. So maybe it's worth to have a closer look with a microscope. Maybe you see other interesting things.
That we gather from certain species. We gather 2 specimens, one for the molecular and one for our herbarium here at the Institute. And when we do, before we put them on the herbarium paper, we do kind of inspect them under the microscope. But I have seen many papers, many scientific articles published on using, looking at the seed morphology and try to differentiate between different ones within seed morphology. They're looking at pollen and differentiating the pollen. So there are studies like that, yes.
Chromosome numbers.
Chromosome number, yes, في كمان.
Okay. So yeah, we can study chromosomes of the species in Palestine. That's another master thesis.
I think it's more than one master thesis.
Okay.
Every general needs a very.
Dr. Isa, if you are still with us.
I think everything is okay. I'm waiting for your questions because you always you have questions. So I'm just waiting your question, Mazen.
Right. Okay, my questions. The first question is, when you mentioned Palestina and this other sister taxon, and you said these data showed that they are different species, or maybe you didn't phrase it this way, but at least that they support that they are two different species. What is a species, and how do you define a species? What is the way that you can know for sure they are different species, or just simply variations or subspecies of the same species? What is this thing about species?
Right, so this is a very, very, there's debate on this question, and different researchers, different scientists have different answers for that. But the baseline, what definition of species is they both specimen need to interbreed together and produce viable and fertile offsprings. And so this is one way. This is a baseline that I don't think people can argue this one. This is one that's not non-negotiable. So they need to interbreed and have viable and fertile offspring. But there's also other things to look at. There's morphology that we need to look at. If they look different morphologically, clearly different morphologically, not a little bit, morphology plays a role. And as we saw in the two Ophrys adonitis and Ophrys palestina, they did look different. One of them was much taller than the other, and one of them flowered 3 weeks after the other one. We also look at the genetical differences, and we did see that in our tree. We saw that they are both two different clades, and the clades even belong to two different ancestors. So they are distinct clearly on the phylogenetic tree that we have. And we already discussed the, being reproductive isolated by if there were two different, if their offsprings didn't have, if they were not viable or fertile, then they have isolation, reproductive isolation. So
my second question is, your husband is here, so you can think what you answer. It's a bit more personal. What do you see yourself professionally? What do you want to do with your career now that you have hopefully done this project and we hopefully publish it? Where are we going, and what do you want to do? Let's say, predict yourself 5 years or 10 years down the road, and tell me, I see Amal doing ABCD.
Amal is happy now in what she's doing. So I see Amal doing what she's doing now, doing research, working with biodiversity, working with the environment. And I want to tackle these questions that we came up with. I want to continue working with research and with orchids and biodiversity. So I would like to see Amal doing what Amal is doing now. بس يعني على level أعلى.
Okay.
I think, Dr. Mazen, to have the ability and the struggle to work with you, I think this is alone, it's a master's degree, because I know you are a hard worker, you are, like Japanese people, you are really hard worker, you do your best that the people who are working with you to work hard like you, appreciate that. And I'm really honored to have some time work with you, because really you work from your heart, not just for the work itself.
Anjad, I want to second what you said, Dr. Walid. Anjad, I mean that with all my heart, not just for this master thesis, but for everything. Dr. Mazen truly pours his heart in everything he does. He never does anything less than the best. And he always expects that of all of us. It's hard, but it's difficult, but it's worth it in the end.
Okay, so having reached this point, any last questions from anybody? If not, what we need to do is have some privacy. Dr. Isa, تفضل.
Sorry for not being able to talk as I am on the way, but I express my sincere appreciation for this excellent quality of work. The work demonstrated a deep understanding of subject matter and high level of analytical skill. Your work will be made a signing contribution to the field of biodiversity conservation and in Palestine. I would like to extend my congratulations to you for your outstanding achievement. Thank you دكتور عيسى، شكرا لك كثير، إن شاء الله نشوفك عن قريب.
دكتور عيسى شو خليتنا احنا نحكم يعني، ما خليتناش شو دكتور عيسى. دكتور رامي يلا نروح هنا.
عارف خلاص. قرار بعدين.
I need the recordings to stop. I need all the people online except stay online. And I need everybody in this room to go outside, get some fresh air for a few minutes while we talk to Rami and